Welcome friends, welcome to the Knowledge Club Online. Today I am going to discuss about the protein synthesis, means translation. All proteins are synthesized according to instructions contained in the DNA nucleotide sequence, which is unique to every individual. Protein synthesis is a two-step process that consists of transcription and translation. At first, during transcription, a molecule of messenger RNA, mRNA, is synthesized according to instructions provided by the DNA. During translation, a polypeptide chain will be produced according to instructions provided by the mRNA. In prokaryotes, transcription and translation occur in the same cellular compartment, the cytosol. Ribosomes are the site of translation. In eukaryotes, not in prokaryotes. mRNA is synthesized in the nucleus from pre-messenger RNA, prem-RNA molecules, and then shifted to the cytoplasm where translation occurs. RNA processing or post-transcriptional modification refers to the molecular mechanisms that lead to the production of mRNA from pre-mRNA. Genes are stretches of nucleotides organized in triplets, a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule, which is called codon. Different arrangements or DNA triplets encode for each one of the 20 amino acids that make proteins. During transcription, a DNA triplet will produce an mRNA codon. During translation, a codon will constitute an amino acid. Like UAA, UAG, UGA, UAA, UAG, AUG, these are the start codon. AUG is the start codon, and UAA, UAG, UGA, these three are the stop codon. Codons are the elongation codon in mRNA. Here we can see the first DNA strand template where the transcription occurs and mRNA is synthesized. And codon, this 3 is a codon, every 3 base pairs making a codon. Then after the translation, the protein is synthesized by coding and a codon-anticodon combination. Before the translation description, I am going to describe about the transcription process. What is it? Transcribed. RNA polymerase binds to the gene's protomer region and starts making a molecule of mRNA until it finds a mark in the gene or termination sequence. The term transcription unit refers to the segment of DNA between the sites of initiation and termination of transcription by RNA polymerase. More than one gene may reside in a transcription unit. In the initiation of transcription, first the RNA polymerase is bind at the start point of the protomer Then the transcription starts, the initiation. Then the elongation of transcription occurred, and at last the RNA is synthesized according to DNA or RNA base pairing rules. A base pair in DNA turns to U in RNA. G base pair in DNA turns to C RNA, as like as T DNA turns to A in RNA. C DNA turns to G in RNA. Where the DNA 5' to 3' sequence is ATGCGTA, GTGGG, ATTAT, it turns to in RNA UAACGC, AU, CACCC, UCC, UAA, UA. Transcription eukaryotic promoters is done in eukaryotes. Promoters are activated by DNA binding proteins or transcription factors. Tata boxes are segments of about 30 base pairs to which RNA polymer is bound. Eukaryotic transcripts pre-mRNA contains exons, coding sequences, and introns, non-coding sequences. Post-transcriptional modification, that is splicing, remove introns before shipping the final mRNA to the cytoplasm. Spliceosomes are organisms in which the excision and splicing reactions that remove introns from pre-mRNA occur. Ribosomes and small nuclear RNAs, snRNAs, are contained in spliceosomes. Next, I am going to the translation process. Every transfer RNA, tRNA, has a specific sequence of nucleotides complementary to an mRNA codon, the anticodon. Opposite to the anticodon, there is an attachment site specific for each of the 20 amino acids. This is the two-dimensional structure of tRNA, where this section is the amino acid attachment site, ACC portion. This is the anticodon, the opposite side present the anticodon, and in between this the hydrogen bonds are present. This is the three-dimensional structure of the tRNA, where it's a L-shaped molecule. One hand it's a present the amino acid attachment site, and in opposite direction it's the anticodon is present. RNA occurs in the ribosomes. Ribosomes have sites of tRNA and anchorage and existing. A growing polypeptide will be produced following instructions in the mRNA. Now, the translation, initiation and elongation of the polypeptide chain in prokaryotes. A start codon AUG complements with the methionine met tRNA in the ribosome, constituting the translation initiation complex. A new anticodon will land in the A site and its amino acid will join met. The tRNA will slide to the P site, leaving the A site free for another anticodon. At first, in initiation, assembly of active ribosome by placing the first mRNA codon AUG or start codon near the P site and pairing it with initiation tRNA, F-met tRNA, which means formamide methionine tRNA, occurs once per polypeptide chain or molecule. And elongation involves 3 distinct steps for each mRNA codon or amino acid in the new polypeptide chain. Transfer of proper amino acid tRNA from cytoplasm to A site of ribosome. Covalent linkage of new amino acid to growing polypeptide chain, peptidyl transfer, movement of tRNA from A site to P site, and simultaneous movement of mRNA by 3 nucleotide translocation occurs multiple times per polypeptide. The elongation cycle is similar in prokaryotes and eukaryotes. First 15 to 20 amino acids added per second, accurate 1 mistake every 10,000 amino acids. Now, how the amino acid tRNA is transferred? Non-initiated amino acid tRNA is placed in the ribosomal A site over the mRNA codon in such a way that a base pairing occurs between the anticodon loop of the tRNA and mRNA codon. Amino acid tRNA transfer is facilitated by 2 soluble protein transfer factors, EFTU and EFTS in eukaryotes. EFTU binds GDP and GTP and is a model G protein, and the EFTS exchanges GDP for GTP on EFTU. The elongation factors are similar in eukaryotes. Instead of 2 proteins, there is a stable trimer. EEF1 means eukaryotic elongation factor 1 alpha beta gamma, which carries out the same function as EFTU and EFTS. EEF1 alpha is the eukaryotic equivalent of EFTU, and EEF1 beta gamma the eukaryotic equivalent of EFTS. EFTU GDP is the inactive form. EFTS activates EFTU by catalyzing the exchange of GDP for GTP. EFTU GTP is the active form which binds to non-initiated tRNAs to which the amino acid group has been attached. EFTU GTP amino acid tRNA is then carried to the ribosome. The complex binds to the ribosome with the amino acid tRNA in the A site. Ribosome binding stimulates GTP hydrolysis, and EFTU GDP dissociates from the ribosome, free to recycle through the step multiple times. EFTU is partially responsible for the high degree of accuracy of protein synthesis via a mechanism called kinetic proofreading. The error rate of protein synthesis is only 1 wrong amino acid for every 10,000 amino acids added to polypeptides. When a charged tRNA is positioned into the A site, the anticodon must base pair with the mRNA codon. If there is an incorrect match, the incorrect AAT RNA dissociates from the ribosome before GTP hydrolysis occurs. If there is a correct match, GTP hydrolysis occurs and EFTU GDP leaves the ribosome before the cognate amino acid tRNA can dissociate, and EFTU GDP dissociates instead, leaving the correct tRNA on the ribosome. EFTU is so important to cellular function that it is one of the most abundant cytoplasmic proteins, greater than 5%. There is one copy of the EFTU protein for each tRNA molecule in the cell. Now it comes to the peptidyl transfer or transpeptidation. The growing polypeptide chain on the P side tRNA, peptidyl tRNA, is covalently linked to the amino acid attached to the A side tRNA. That is, a peptide bond is formed. The P side tRNA is now unacylated and the A side tRNA is covalently linked to the growing polypeptide chain. This step is somewhat of a mystery but is believed to be catalyzed by a region of the 50S subunit called the Peptidyl Transferase complex. The complex utilizes both proteins and 23S rRNA. There is growing evidence that 23S rRNA actively catalyzes the peptidyl transfer step, but the mode of action is yet unknown. Now it comes to the translocation process. The A side tRNA with the growing polypeptide chain is then moved within the ribosome to the P side, while the deacetylated P side tRNA is moved to the E side. Simultaneously, the mRNA is shifted by exactly 3 ribonucleotides or 1 codon. The process requires a soluble protein factor called elongation factor EFG. EFG is similar but much larger than EFTU. EFG is also active as the GTP complex. Although many older text reports that the binding of EFG-GTP to the ribosome provides the energy for the translocation, it is now known to be wrong. The energy derived from GTP hydrolysis drives translocation. After GTP hydrolysis, EFG-GTP is released from the ribosome. The tRNA carrying the polypeptide chain and the next mRNA codon is in the A side. The ribosome is now ready to start the elongation cycle over again to add a new amino acid until a termination signal is reached. It appears that EFG catalyzes its own exchange of GDP and GTP. No soluble guanine nucleotide exchange factors has yet been found. The equivalent EFG in eukaryotes is EEF2. Now comes to the termination of translation. A stop codon UAG, UAA or UGA signals the end of the mRNA molecule. A release factor triggers the disassembling of the 2 ribosomal units and the mRNA molecule. Soluble protein release factors RF1 responds to UAA or UAG, RF2 responds to UAA or UGA, RF3 are GTPase like EFTU and binds in a similar A side location. RF1 or RF2 interact with RF3 GTP, have a similar step as EFTU GTP, A tRNA or EFG, and bind in a similar ribosomal site A site in a manner similar to EFG. GTP hydrolysis drives the movement of the terminal mRNA codon into the P site, moving the last tRNA into the E site and off. At the same time, the polypeptide chain is released after hydrolysis of the tRNA peptide bond. In eukaryotes, only a single release factor ERF is necessary. It recognizes all three stop codons and interacts with GTP. A mutation resulting in a premature stop codon is called a nonsense mutation. Now, the polyribosomes. An mRNA molecule is generally translated simultaneously by several ribosomes that constitute polyribosomes. Next, after the protein synthesis or protein translation, how the protein is transferred or translocated? It's translocated by the vesicle formation or the coat protein formation from the COP1. It transfers to the from the endoplasmic reticulum to the Golgi apparatus by the forming of coat protein. Maybe this lecture is helpful to you. Thank you.